Good morning, everyone. So I was asked to spend a few minutes speaking about the environmental component or dimension of antibiotic resistance, and I'll wrap up my talk with one or two comments about this within the context of surveillance. So what I'm going to say is going to reiterate a number of things that we've heard this morning. So the One Health framework has three components: people, animals, and the environment within which these creatures live. We live in the other creatures. Why is the environmental dimension of AMR potentially important? A couple of reasons. Where do AMR genes in human pathogens come from? We think they come from microorganisms from the pangenome of the microbial world. Where are those microorganisms? They are in the environment. So determinants of resistance that end up in human or animal pathogens are recruited from that environmental reservoir, if you will. We think that humans are modifying that environmental reservoir in an undesirable way by increasing the abundance of antibiotic resistance genes, by increasing the mobility of those genes, by increasing the suite of antibiotics that resistance can be found within that reservoir. So that's one thing, the so-called resistome. And the second element, again, as we've heard a number of times this morning, is that these resistance determinants can be transmitted between people and animals, between people and people, via the environment, particularly through waterborne transmission. And so that is one component of the One Health framework. To put that in a more complicated way, and this is a figure that Rebecca is largely responsible for, there's kind of three parallel conversations that go on with respect to managing, mitigating AMR. And what I think of One Health is simply that these three conversations have to be within the same room. So one conversation has to do with human medicine and how we manage antibiotics within human medicine, how transmission happens between people and people. The second conversation is other creatures, and these creatures are found within terrestrial food production systems, aquatic food production systems, and companion animals that people generally don't speak about, but we do give drugs to our dogs and cats. So the second conversation is how do we manage antimicrobial resistance with other creatures? And the third conversation is the environment. And the concern is largely that antimicrobial resistant bacteria that are selected for in humans or in these other creatures are transmitted into the environment through waste streams, contaminate the environment, change the environment, and then we are exposed to organisms that are transmitted via the environment. What is the evidence that we're doing something to the aquatic environment that we need to worry about? There's a large body of evidence looking, for example, at wastewater treatment effluents, so municipal wastewater treatment plants, the effluents that are going into aquatic receiving bodies. Those effluents contain bacteria, genes that confer resistance, antibiotic residues. We are putting these things into the environment, we know that. And so there's a large body of literature concerning surface water, fresh water, and marine systems, and also a number of studies, for example, this Canadian one, showing that drinking water, so groundwater, can be contaminated with these organisms, particularly in areas where there's a lot of food animal production. What's the evidence that we're doing something to terrestrial environment that we need to worry about? Again, this has to do largely with waste streams going into the terrestrial environment, particularly with respect to agriculture. So if we give drugs to animals, we enrich for resistance in those animals. When we put the fecal material of those animals onto ground that we use to produce crops, we entrain those bacteria, we entrain those antibiotic residues. In some parts of the world, including in Ontario, we feed drugs to people. Those people excrete those drugs and the bacteria that were selected for in those people, and we apply that sludge into crop production systems. In Ontario, for example, population of 13 million people, the waste from roughly 60% of those people is recycled onto ground. That varies according to where we are in the country. The good news is that this offers opportunity to manage that, so we can regulate the use of drugs in food animal production. We can, in many production systems, the waste is stored for some period of time, so we can treat it in that way. And we can regulate where and when and how these materials are applied into crop production systems. What is the evidence to substantiate what I just told you? Many studies showing that, this particular one from China, showing that fecal material from farm animals that are medicated contain large numbers of antibiotic-resistant bacteria and genes, and that crop production systems that receive these materials are enriched in antibiotic-resistant bacteria. And in fact, we can show in many instances a transfer of bacteria from manured ground to the crops that are harvested from that ground, which is another potential transmission route to people or to animals that are grazing that ground. There is a concern that various anthropogenic activities are doing something to the resistome that's undesirable. One part of that is the potential that we're accelerating bacterial evolution. So, for example, we know that under selection pressure of antibiotics, we can accelerate the rate at which phage move genes around. We can increase mutation rates by downregulating DNA repair systems. And we can show that in aquatic systems that received wastewater, a very pronounced increase in abundance of mobile genetic elements or elements that can move genes around. And so that is a concern. When you think about the environment, One Health, at what scale should you think about, and at what scale should you think about mitigating AMR through the environment? So if we clean up our act in Canada, is that going to be protective of us? In fact, it likely won't be, and one of the concerns has to do with, for example, the NDM1 gene, which was thought to originate from India, then distributed into the UK, then various other places through mobility of people, probably people who went into India for medical reasons. But a recent study showed, interestingly, that in New Delhi, when they sampled creeks, streams, ponds, you name it, they found a very wide distribution of a variety of microorganisms that carry the NDM1 gene. So the idea here is that people in New Delhi that live within that environment are being constantly exposed to bacteria that carry this gene, then people come from the West and pick it up. So in low-income countries where there's a very poor infrastructure for wastewater treatment and sanitation, that has a consequence for us in the West. And so when you think about the environment, AMR, you need to think about it globally. You need to think about solutions that are appropriate for high-income countries, but also low and medium-income countries. So how does this fit into surveillance and what one might want to do in surveillance? Broadly speaking, there is no systematic environmental surveillance, either in this country or internationally, that I'm aware of. There are, with one or two exceptions, one exception in Canada would be that the food net sites, the Sentinel sites, do include water in their surveillance program. Yep, less than 2 minutes. And what we know about AMR in the environment generally comes from ad hoc research programs that are undertaken by government agencies or academic colleagues. So overall, there's a lack of surveillance data that would be very useful or needed to establish the significance of environmental transmission routes relative to other routes like foodborne transmission, undertake a human health risk assessment for environmental AMR, understand the health significance of the anthropogenic impacts that I've mentioned, and use this information to inform policy decisions on best practice for managing waste streams and the relative value of investments relative to other steps that one might want to take to protect people from AMR bacteria. And with that, thank you.